Today I will again discuss on Oedogonium. So on that day we had already discussed on the thallus structure, then the cell structure, okay, as well as the growth and cell division also we had discussed the in that previous video. Now we will start with reproduction, okay, reproduction in Oedogonium. So as you can see that uh, actually Oedogonium, it can reproduce by vegetative reproduction, asexual as well as sexual reproduction. So we are going to start with the first one, that is vegetative reproduction. Now in vegetative reproduction, the vegetative multiplication, it takes place by either of these two methods, that is fragmentation and akinetes. Okay, so what happened in fragmentation, like many other algae, the small fragments, okay, there are small fragments of Oedogonium filament. They have the capability to grow into complete filaments under favorable condition. Okay, so in here in these there are small fragments. In case of fragmentation process, these small fragments of Oedogonium, they have the capability to grow into the larger fragments, into complete fragments under favorable conditions. If the conditions are favorable for the growth, they are going to grow into complete filaments. And actually the fragmentation process, okay, in vegetative reproduction, it takes place by either the accidental breaking of either of these filaments, the intercalary cells, okay, of the accidental breaking, or it may be due to the dying of these fragments, or due to dehydration of the intercalary cells. So due to this process, the small fragments are formed, and on the favorable condition, those small fragments, they will grow into complete major fragments. Okay, so that is fragmentation. That is reproduction through fragmentation. Okay, now the next one is the akinetes. These akinetes, they are actually the thick-walled reddish or brownish structures. Okay, they are reddish or brownish structures which are usually formed in small chains, as you can see here, chains of akinetes. Okay, they usually form in small chains during favorable condition. During when the condition is unfavorable, then these akinetes will start to form, and they will germinate only when the condition is favorable, and they will form new filaments. So the vegetative reproduction in Oedogonium, it can occur through fragmentation or through the formation of these akinetes, which are formed during under unfavorable condition, but they will germinate when the condition is favorable. Now next we come to the asexual reproduction. Now here the asexual reproduction, it usually occurs by the means of zoospores. Okay, so by means of zoospores, the asexual reproduction, it occurs. And these zoospores are, as we know, they are the spores which are capable of swimming, the zoospores, okay, by means of flagella. And the zoospores, as you can see here in this figure, they usually form singly. Okay, only a single zoospore is formed within a cell. Now usually the cell with the apical cap, okay, the cell which has the apical cap, it can behave as a zoosporangium. Alright, so just remember that the cell which in asexual reproduction, what happened, the cell which has the apical cap, it will behave as a zoosporangium, that is the asexual structure in which the zoospore will form. Alright, so this asexual structure, it will form in those cells with the apical cap. And during the zoospore formation, what happened during the zoospore formation, the cell contents, they will contract slightly. Alright, the cell contents, they will contract slightly from the cell wall. They will contract slightly from the cell wall, and actually what happened, a small lens-shaped hyaline area. A small lens-shaped hyaline area, it develops on one side of the protoplasm, which eventually becomes the anterior end of the zoospore. Okay, so you can see this part here actually. This part here is the hyaline area. Okay, that is the translucent area which will act as the anterior end of the zoospore. Okay, so you can see in figure C here, the glassy or translucent area here, the hyaline area, will develop on one side of the protoplasm. You can see this whole protoplasm here. Okay, in this cell, first the cell will act as the cell with a cap will act as a zoosporangium, then the protoplasm, okay, the formation, the cell contents will contract slightly. As you can see here, it already contracted, and a small lens-shaped area, hyaline area, translucent area here will develop, which will eventually become the anterior end of the zoospore. Now at the base of this hyaline area, what happened? At the base of this hyaline area, a ring of granules, okay, a basal granules will appear. A ring of basal granules will appear, and from each of these granules, flagella will form. A single flagellum arises, and the basal granules, it remained connected with each other by the fibrous strands. Okay, they remain connected with each other by fibrous strands. And if you take a look at the mature zoospore, when the zoospore is formed, you can see that the zoospore is ovoid. Okay, it is ovoid, then it is spherical, or it is pyriform. As you can see, pyriform meaning pear-shaped. Okay, so it is either ovoid, spherical, or pyriform, and also it is uninucleate. You can see a single nucleus is present, so it is uninucleate and also contains chloroplast. Alright, so sometimes actually the zoospores here, it has an eye spot. Okay, just remember that sometimes the zoospore, it has an eye spot. An eye spot, it is an organelle, a photoreceptive organelle. Alright, and this photoreceptive organelle, it helps the cell to sense the light direction or the intensity of the light and respond to it. So that is the function of the eye spot. So zoospores, sometimes they also possess the eye spot. And at maturity, what happened? At maturity of the zoospore, the wall of the zoosporangium, it will split near the cap region. Okay, the wall will split near the cap region, and the adjacent cell, it moves apart to make a passage. Okay, the adjacent cell, it will move apart to make a passage for the liberation of this zoospore, for the liberation to the outside environment. Alright, so it is believed that the mucilaginous substance is secreted at the base of the zoospore in sporangium. So here inside the sporangium, it is believed that a mucilaginous substance is secreted. That is, a mucilaginous substance, a substance which has a a gelatinous consistency or a viscous consistency, alright, a viscous substance or a gelatinous substance will be secreted in the zoosporangium at the base of the zoospore, which aids in the extrusion of the zoospore after absorbing water. Alright, it will export this zoospore will be export outside the zoosporangium after it absorbs water. And the zoospores, after it comes out of the zoosporangium in a delicate mucilaginous vesicle, as you can see here, the presence of a vesicle, that is an air-filled swelling in a plant. You can in this algae you can see this is an air-filled swelling, okay, the vesicle. Then along with this, the zoospore will come out from the sporangium, okay, will come out of the sporangium in a delicate mucilaginous vesicle. And this vesicle, it will soon get dissolved and the zoospore is liberated. So you can see that the zoospore now is liberated outside. Now after liberation, what happened? The zoospore will start to germinate. The zoospore, actually it will first swim for about an hour, and then it will settles on any solid substratum. Alright, substratum, it can be anything, as I had mentioned in those previous videos. Alright, it can be a dead plant, a rock, a tree, or anything. Okay, it will attach to a solid substratum with its anterior end out downwards. Alright, so it will attach through its anterior end to the substratum. And during that time, when it swims for an hour, it will attach to the substratum with its anterior end downwards, and it will retract its flagella. Okay, it will retract its flagella and it will start to elongate considerably. As you can see here in figure F, it starts to elongate, figure F and figure G. So from E, F and G, you can see the difference. Now a transverse septum, okay, now you can see here after it starts to elongate, what happened in figure G, septum will start to form, which will separate the base hyaline holdfast. Remember in the previous video I had mentioned what is this holdfast. So this septum which form here, it will separate this hyaline holdfast from the apical green cell. Alright, from this green cell. And what happened to this green cell? This cell will start to divide repeatedly to form a new filament. So you can see this is the whole thallus structure of Oedogonium. So it already started to form a new, it will divide repeatedly to give rise to a new thallus structure, to a new organism, okay, to a new filament.